Hello everyone, my name is Jamal Mosleh. I'm CEO of Elysera Therapeutics, which is a Swedish cell and gene therapy company developing next generation armed CAR T-cell therapies and oncolytic viruses. On the picture here you see Professor Magnus Sand and his research colleague DU, and they have together with their team developed two CAR T therapies, two oncolytic viruses, and a platform technology we call iTank, that can be used to arm CAR T-cell therapies in order to help them to meet two of the major challenges that these therapies face in the treatment of solid tumors. This is a slide presenting our project pipeline. You see that we have 4 programs under development. ELC-301 is a CD20-directed CAR T therapy armed with the iTank platform that is soon to enter clinical trials. So we have secured funding and also the regulatory approvals to initiate a clinical study in B-cell lymphoma. We call this study the KARMA trial, and we intend to treat the first patient right after summer vacations here in Sweden, so in August. ELC-401 is also a CAR T therapy. This time we are targeting IL-13RA2, which is a target that is overexpressed in glioblastoma, but also a target which we think is overexpressed in a number of other solid tumors. So there's a potential to broaden the portfolio further if we or a potential partner should choose to. ELC-401 is in preclinical stage, as is 201, but we are preparing both of these programs for clinical studies, and we intend to enter clinical studies either with a partner or through soft funding. So we have different initiatives for both of these tracks. ELC-201 is what we call a next-generation oncolytic virus. It's in theory applicable for treatment of any solid tumor. It's armed with the iTank platform and another immune-stimulating factor to trigger a parallel immune response against cancer. And ELC-100 is the fourth and last program, and actually the only candidate which we haven't armed with the iTank platform. But this is an oncolytic virus which we have developed specifically for treatment of neuroendocrine tumors, and we have an ongoing clinical study in 12 patients. We have treated 11 out of 12 so far, so we only have 1 patient left to treat during this year. Talking about CAR T-cell therapies, it's worth to mention that CAR T-cell therapies lies within the immuno-oncology space. And of course, in immuno-oncology, you can use the patient's immune system in many different ways to treat cancer. And I would say that CAR T-cell therapies have been shown to perhaps be one of the most promising types of immuno-oncology out there. And the reason I say this is because we have now long-term follow-up studies showing patients that were treated with CAR T therapies over 10 years ago, and you follow them up and you see that they have an intact immune system against cancer, and they still remain tumor-free, which shows the curative potential of these types of therapies. However, you only have approved CAR T therapies in the space of hematology, blood cancer. 6 approved therapies. So no CAR T therapy has yet to be approved in the solid tumor space. And of course, the solid tumor space is much larger, about 10 times larger. That's where you have the highest unmet medical need and of course also the highest commercial potential. So why don't CAR T therapies work as well in solid tumors as they do in blood cancer? And there are 2 main reasons for this. One, solid tumor has a very heterogeneous antigen expression, meaning that it's difficult to find a target in a solid tumor which is overexpressed on all tumor cells and not just some of the tumor cells. You don't have this problem in blood cancer. And in this picture here, we're trying to exemplify that by these blue cells here, showing the tumor cells that lack the CAR antigen target and therefore will not be attacked by the CAR T-cell therapy, so they will form CAR T-resistant metastasis they are no longer able to treat. The second big challenge that CAR T therapies face in treatment of solid tumors is that you have a very immunosuppressive tumor microenvironment in a solid tumor, which you don't have in blood cancer. And this immunosuppressive tumor microenvironment counteracts the CAR T therapy, it exhausts the T cells, and it also makes it difficult for the CAR Ts to penetrate the tumor and come into contact with the tumor cells to kill them. And both of these challenges we believe that we can meet with the I-Tank platform. So due to a short time here to present this, I won't go into the details on the mode of action of the I-Tank platform, but just rather tell you that the point of the I-Tank platform is to trigger a bystander immune response. The point is to have a parallel attack on cancer in parallel to your CAR-T therapy. So what we're trying to do with the I-Tank platform is to trigger the patient's own T cells against multiple targets on the tumor cell, not just the target that the CAR is directed towards. And what the I-Tank platform also does is that it creates an environment that directly counteracts the immunosuppressive tumor marker environment in solid tumors, and which actually also strengthens the function of the CAR T cells. Now, of course, we use this platform technology in our own candidates, but you can actually use it in basically any type of CAR T cell out there, regardless of which antigen target you are going after or which other potential genetic modifications you've done to your therapy. It should be able to add the I-Tank platform to these CAR T therapies. So that's why we have a little bit of a different business model for the I-Tank platform in comparison to our other drug candidates. Ultimately, we intend to license all of our programs and assets, but for the I-Tank platform, we intend to license this multiple times to be able to capitalize on this in several licensing deals, generating upfront payments, milestone payments, and potential royalties. And I use this slide to show the potential and a benchmark for both for the I-Tank platform, but also for our other CAR-T candidates. The first example here is from a Japanese company, which is actually the only other company, and I'm sure there are others out there, but we haven't identified them, that has a similar platform technology that they call Prime-T. And they're basically aiming to do what we're aiming to do with the I-Tank platform. And this Japanese company has been able to successfully license their technology multiple times to both big pharma companies, big biotech, and smaller biotech companies interested in the field of CAR-T therapies. And we believe we can do the same thing. The example in the middle here, Novartis licensed a CAR-T candidate from Legend. The reason this example is relevant for Elysera is because ELC-401, which we are developing against glioblastoma, is in preclinical phase. It's an armed CAR-T therapy, and it's possible to broaden this further into other indications, just as Legend's candidate. So a preclinical CAR-T candidate armed with a technology that, unfortunately, we don't have more information on, but generated $100 million upfront. And the last example here is from Janssen, which licensed two CD20-directed CAR-T therapies from a Chinese company called CBMG, now called Abelzeta. They had performed two initial clinical studies, not that unsimilar to what we're aiming to do with the KarMMa study. Of course, very nice results from this study, generating $245 million upfront. So we believe that all of these different examples can be related to what we're doing at Elysera and shows the level of interest that the industry puts in these types of therapies. Now we have reached preclinical proof of concept for the I-Tank platform, and we've published extensive preclinical data in Nature Biomedical Engineering, which can be considered one of the more prominent scientific journals in the world. There we can show a broad attack on multiple targets when we compare I-Tank armed CAR T cells with the efficacy of unarmed CAR T cell therapies in different mouse studies. So regardless of which mouse model we use, which tumor target we're going after, or which indication we're going after, we see a prolonged survival and lower tumor growth when the mice are treated with I-Tank armed CAR T cell therapies. Which also indicates that this is universally applicable with any CAR-T treatment. And also important to mention here is that adding the I-Tank platform to your CAR-T therapy doesn't add any toxicity. So it's equally safe or dangerous to use the I-Tank armed CAR-T therapies as it is to use your conventional unarmed CAR-T therapies. The KarMMa study that we've planned for ELC-301 in B-cell lymphoma is targeting CD20 in contrast to CD19, which the three approved CAR-T therapies today in this indication are targeting. So there are already approved therapies in this indication, but the point here is that 50% of the patients relapse and are in need of additional treatments, and this is the patient population that we primarily are going after in this case. The plan is to treat in total 18 patients, 12 of them in the dose escalation phase, 6 patients in a dose expansion phase. In total, we will have 12 patients receiving maximum tolerable dose, which we believe is an adequate number of patients to draw some conclusions about efficacy and, of course, also safety. And substantial soft funding has gone into this trial, around 40 million SEK. So we have funding to recruit and treat all 18 patients. We intend to report data from the first cohort of 3 patients by the end of this year, and from the first 12 patients by the end of 2025. Looking at the ELC-401 and glioblastoma, we believe that this is a good indication to target for this specific candidate for the reason that, one, the target that we're using has reached proof of concept in this specific indication, so we know it's a relevant target for glioblastoma. And also glioblastoma is has a very heterogeneous antigen expression, meaning that patients that respond to CAR T therapies very quickly get relapses. And it's also a very aggressive tumor with a very aggressive tumor microenvironment. So adding the ITANK platform to a CAR T therapy, specifically in glioblastoma, makes a lot of sense. We have planned a clinical study with this candidate. We have produced the viral vectors that we need for the CAR T construct. What we don't have is funding to initiate the trial, but we have different soft funding applications that we both intend to submit and also that we have submitted. So we're hoping for some positive news from those. And we're also talking to potential partners to also have that as an alternative to move this into clinical studies. The 2 oncolytic virus programs that we have, ELC-100 and ELC-201, are somewhat different. So ELC-100 is specifically developed for treatment of neuroendocrine tumors, while 201 you can use to treat basically any cancer. And in contrast to ELC-100, ELC-201 is armed with the ITANK platform and another immune-stimulating factor to really trigger this bystander immune response. ELC-100 is, as I mentioned, in a clinical study, fully financed by VictoryNet Foundation, which is a foundation focused on research and development, specifically in neuroendocrine tumors. The milestones that we have ahead of us the coming 12 months are a lot with a focus on clinical milestones. So it's an exciting year we have ahead of us. So first off, we will start the KARMA study, as I mentioned in the beginning of, at the end of this summer, in August. And the first patient cohort data report end of this year. And we only have 1 patient to treat in the ELC-100 study, so that patient will hopefully be recruited this year with a full data report in 2024 as well. And we will come back and report to the market how we intend to continue developing this program based on the data that we collect after completing the study. And then for the ITANK, ELC-401 and 201, we have several potential news that I can't promise that we will reach them, but we're working intensively to reach those milestones as well. So we are already in discussions with potential partners for licensing the ITANK platform. Hopefully some positive news from that track sometime during this year. And as I mentioned, for 401 and 201, we don't intend to finance clinical studies on our own if it's not with soft funding or together with a partner. And we're working to secure that as well. So with that, thank you so much, and
thank you, Jamal.
Ask some questions.
Thanks a lot, Jamal. A great presentation. Unfortunately, we don't have much time. I'll go with one question here. You talked a lot about the recent deals that there have been in this field with CAR T cells and also technologies similar to ITANK. How is ITANK compared to, I want to say better, but compared to these other technologies that have just gone for some major deals with Big Pharma?
So the iTank platform is a arming technology for CAR T therapies. And of course, we're not the only ones that have thought that this is important. But I think that we are one of the few that actually has something that could be a solution to the problems that CAR T therapies face. So if you ask me about Legend Biotech, for example, I can't tell you the difference between our technology and theirs because they are not releasing more information about that. The same was with, for example, AstraZeneca also has an arming technology, which you have very little information about. But looking at recent press releases from Astra, for example, you can tell that their arming technology is based on targeting the immunosuppressive tumor microenvironment in solid tumors. So in that sense, it's somewhat similar to what we're doing, but not all the way to also helping the CAR T to face the challenge of antigen heterogeneity. The Prime T platform from this Japanese company, however, they can claim that they are doing both of these challenges, meeting them. The difference is that they are using 2 immune-stimulating substances to reach these goals, while we are going a different direction, and we're using a protein from a bacteria. And the point with using a protein from a bacteria is to really trigger an immune response from the patient. So in a sense, you can say that using the iTank platform is like using an adjuvant to create like a therapeutic cancer vaccination in combination with your CAR T therapy. So this bacterial-derived protein will basically trigger an immune response that helps the patient's own T cells to target multiple targets on the tumor cell and also counteract the immunosuppressive tumor microenvironment. So both of them at the same time, basically.
All right. Well, thank you, Jamal. Unfortunately, that's all the time we have for questions. But if you stick around for lunch, hopefully some other people can ask you some more questions.
Thank you.
Thank you.